really cool biochemistry question that you want to answer, how are you going to approach actually answering it? You can do things in vitro, either with like a reconstituted or a lysate-based system. You can do things in cells, you can do things in tissues, you can do things in vivo. Each of these has different pros and cons, and today I want to go over some of the things to think about when you're trying to determine which of these experimental systems to use for your experiment. So first, some of the terminology, and note that sometimes people will use the terminology differently, and sometimes there's a little bit of a gray area. But we can think about things being two extremes being in vitro and in vivo. So in vitro is going to be like outside of cells. So sometimes this is called like in a test tube, but it doesn't actually have to be in a test tube. It just has to be outside of the cellular and organismal context. We are going to further go into different types of in vitro. So doing everything from scratch, like we're constituting it, or doing it based on a lysate. So basically splitting cells open, maybe adding stuff to what was there, but doing things in this cell-free context, but based on what was inside the cell to start with. At the other extreme, we have in vivo. And so this is going to be happening inside of organisms. And so one of the benefits about the in vitro was that because it was outside of the cells, we could control things better. So we had the most control. When we're talking about in vivo, here we have the least experimental control, but it's more realistic. Well, at least for the organism that you studied. So if you're studying things in a mouse, that's not necessarily directly applicable to like humans. And so you have to keep that in mind. And the middle, we have cell culture. So this is like a medium level of control and realism, more variability. Some people refer to cell culture work as in vivo, but it's not really unless you are working with a unicellular organism. So if you're working with bacteria and yeast, then cell culture and like in vivo is the same thing because these are these single-celled organisms that you're working with. But when you're working with cell culture from like a larger organism, plant or an animal or something, then that's not really in vivo. There's also some like in the middle stuff. We can talk about organoids. We'll go more into what those are in later, but basically you take cells and you grow them more in like this clumpy type thing where you can even have multiple cell types, and you get like the extra cellular environments and various things to make it like more realistic and tissue-like, and then you can have actual tissues. And you can also do things in like organs and stuff. But so we have, when the more realistic you make it, the least control you have, but and also the more variability you introduce because organisms are variable. And so this variability can be important, but it can also be misleading. And so you often have to use different cell lines, different organism, almost different strains, various things to try to make sure that the findings that you find are actually generally applicable. So we'll get into more of these things, but this is just a general overview. So let's start with the in vitro and experimental systems. So this is what I love. I love working in vitro because you have the most control and you don't have to work with animals or anything. This is really good for dissecting mechanisms. So trying to figure out how things work. And it's good for this because you have the most experimental control. You can do things like easily introducing labeled components, and you can easily introduce compounds that might not be easily able to be introduced into cells or into cells inside of organisms. So this can be things like initial hits for pharmaceutical drugs. So like before they optimize them, they often do like screens to find like potential hit compounds and then further optimize them, making them more like a bioavailable so that when they get into the body, they're not just going to get broken down or excreted, and they're actually going to get into the bloodstream and then into the cells and that sort of thing. And so there's a lot of optimization that goes on. And before that optimization, the compound, it might have activity towards the whatever it's trying to do, but it won't actually be able to get into the cells. But when you don't have cells to get into, things are just like in a lysate, you can then add that compound directly.
So with the experimental in vitro systems, there are decisions you have to make. And one of the main ones you have to make is whether you're going to do a reconstituted system, like from scratch, or lysate-based. So with the reconstituted, basically you're making it from scratch. You're purifying the individual components if necessary and mixing them together, typically using only what is necessary and sufficient for the reaction to occur. And then you can do things like make changes to it, introduce different things, and see how that changes the system. With a lysate-based mechanism, you break cells open, so you lyse them, you remove the membrane bits, so you can like centrifuge it to spin out the heavy membrane, leaving you with the soluble stuff, and then you can use that remaining lysate. Either you can partially purify it, or you can just use it unpurified. And you can use that as a reaction component along with additives particular to your experimental question. So later we'll get more into an example, a practical example of this is something that I've been thinking about a lot. It's a Selfie protein expression. So making protein outside of cells, and this can be done either with like reconstituted from purified components like this pure system, or based on in a lysate-based system. So with a reconstituted system, pros is you have the most control. You know exactly what you're putting in there. You can control the concentrations of what you're putting in there. You can make sure that nothing's in there that you don't want in there. Various things like that. You can easily alter the concentration, and you can easily introduce mutated variants without having to do genetic engineering in cells or anything. So when you're using one of these reconstituted systems, when there are protein components that you want to introduce or RNA components that you want to introduce. So with the RNA, you would do like in vitro transcription. With the proteins, you can do like use recombinant protein expression and then purification. And I've talked about this a lot in other posts. But you can basically introduce the instructions for making a protein, get cells to make the protein, then purify out the protein. Because you're giving them the instructions for making the protein, you can give them modified instructions, and then they'll make a modified protein, and then you can test how that protein acts. So very cool, and easy to swap that out in one of these reconstituted systems, as long as it's like a system that you've made yourself. If you're buying a commercial system, it might be hard, because if things come as a mix, you can't just like do that easily if it's already in the mixture. Because you have that control, you can have the best consistency. You don't have to worry about batch to batch variability with your lysate, cell lines to cell line variability, passage to passage when you're passaging cells. Like you grow them, you split them so they're more media because they're growing, and then you split them and it's called like you passaging them and stuff. And then you can introduce more variability because you have the chance for like mutations to occur and that sort of thing. But when you're doing this in vitro reconstitution, then you don't have to worry about this sort of things. But there are things that you need to worry about. So the most is that it's the least realistic. There might be components that are missing that aren't necessary for the reaction to occur, but they help the reaction happen in vivo. So they're actually normally there, and they help things happen, and they might even make things happen in a different way than would happen here. And so you could get kind of led astray if you're trying to figure things out, but you're missing a component. So you might have to purify a lot of different components. And so recombinant expression, where we put the gene, as we were talking about, we put the gene into cells and get those cells to make it for us, as well as in vitro transcription, where we can make RNA in our test tube based on a DNA template. These have made it an easier task than if you had to purify the native protein, so purify them like out of cells that were like the cells that they're normally made in, but there might be a lot more of components that you need to express and purify. So even though it's easier, it's still not easy. Okay, so what about a lysate-based? This is going to be more realistic than with the reconstituted, because it's going to contain components that you aren't even sure what were, but they were in the cells and they enable this. They might make this process enhance the process or make it happen in a more like realistic way. A pro is that you can still add components that aren't present. You can still add drugs and that sort of thing. And you can add more of components that are present, but not in high enough concentrations for your purposes. So often like inside of cells, there are going to be things that are kind of like compartmentalized, or they're going to be scaffolded together or somehow. So you're like increasing local concentrations of things. And so if there's a process that's happening, it's going to be happening in this more like concentrated area of the cell. Whereas in a lysate, everything's kind of just more diffuse and spread out and lower concentrations. So if you're just having the lysate, you just want it to do that one thing, and as opposed to all the stuff that it normally does, give it more of the stuff that it needs to do that one thing, such as like make a protein, and then it'll have better efficiency of doing that one thing. And so we'll look at this when we talk about the cell-free protein expression, how you add amino acids, you add tRNA synthetases, and that sort of thing. So you still have cons, and you have cons compared to the reconstituted is that you have less control than with the reconstituted. You don't even know the precise concentrations of things. If something was in the lysate already, then you can't change it unless you change the cell type or you somehow remove the components that were in the lysate, such as by immunoprecipitating out the wild type version of the protein, and then that'll tell you if the protein's actually needed for the thing. And or you could replace it with a mutant form that you expressed and purified separately. And so by like immunoprecipitating out, basically you can take, for example, if you had like beads that were conjugated or attached to an antibody against the thing you want to get rid of, and then you can kind of like pull it out, or you can and that sort of thing to remove that thing that you don't want. But you do have batch to batch variability because you're taking things out of cells, and cells have variability. So with these in vitro experimental systems in general, both of the methods, this is basically like the least maintenance required. So getting it up and running can be very daunting, especially with the reconstituted system. So there's a big upfront investment. But if you're able to purify enough of the components to last a long time, and if they store stably, then you can do a lot of experiments without having to do any more work. So you don't have to feed and passage the cells or that sort of thing. And so you have the quickest ability to test a question once the system is up and running. You can do like an experiment that'll take a couple hours or so instead of a cell-based experiment that's going to take you a longer term time. So you can test things more quickly, but it's going to be the least realistic, as we mentioned. You may be missing components. We don't have that compartmentalization as I talked about, so you can add components to increase the concentration, but the compartmentalization and the scaffolding proteins and that sort of thing that might actually be important for having that process happen normally. Another thing that is that if you want to study like membranous proteins or systems, you might need to consider things like liposomes if you're doing an artificial system. These are kind of these like lipidy bubbles that you could use to have proteins embedded in a membrane context. And then, of course, the results might not be physiologically relevant if you get the system up and running. If you're testing you can find answers, but is that actually relevant? Like if you're testing a drug that can't even make it into cells, and or that it's getting like degraded before it gets into cells, what should you make of the results? And so if you're trying to answer those sort of questions, you might want to look to a different system. But it can be good for getting those initial like mechanism-based things. So when you're doing a reconstituted system, you're gonna have to make some decisions. And so one of them is going to be like, what are you going to make yourself versus purchase? So this gets into, well, first of all, what can you purchase? So what's commercially available? Even if it is commercially available, is it simple enough to make yourself? And is it worth it to make it yourself? So sometimes you think, oh man, that's so expensive. But these companies, they make a lot of it. And so if you were to do it yourself, it wouldn't be, basically if you don't need very much of it, these companies are going to be making it like in bulk. And so they're selling you at a price that is going to be, it might seem expensive to you, and it's a profit for them, but it's cheaper than if you were to actually make it yourself because you don't need it at like bulk scale. It might also take you a long time to make it, a lot of resources, maybe you don't even have those, a lot of time and energy, and it's just not worth it. So some things you're going to want to purchase when they are commercially available, but be wary that if you're using like a kit, things might be mixed together, then you'll have less control. So you also might have to consider like what organism components are you going to use? So with the protein expression system, are you going to use the E. coli aminoacetylDNA synthetases, or are you going to use like the homologous human version? So the human version, the homologous one, so like basically like the same thing, but in the human form. And so often published protocols, they're kind of like mixing and match where components come from without really offering an explanation for why. So sometimes it might be important. So sometimes like one species version works more or better, more efficiently, or works better in vitro. Maybe it doesn't require cofactors or other homologs as other homologs would. And sometimes it's just because that's what they had on hand, and or that's what people had used in the past and want to fix it when it isn't broken. But you might want to experiment with different sources and might have better luck sticking to a single organism where everything is maximally compatible with one another. And this is going to depend too on what your goal is. Is your goal to use this system just like to make protein, say, or is it to actually study how proteins get made, in which case you might want to really be more concerned about having everything be as close to realistic as possible. Whereas if you were just going for like maximal protein production, then you might just want to be going with what's most efficient. Okay, so if you're going with a lysate-based system, here your decision is mainly going to be like, what type of cells are you going to get the lysate from? Are there special things that you need to do when you're harvesting them and preparing the lysate to make sure that the lysate's like competent for your reaction? So you need to make sure that you're preparing this lysate in a way that's preserving whatever sort of activities in the lysate that you want to preserve. So look to publish protocols and follow them, at least to start with. Make sure that you can recreate what they did before you try to modify things too much, or else you might end up not knowing what you need to troubleshoot. And so an example is the Selfie protein expression. So this can be done with a reconstituted or a lysate-based system. And so a reconstituted system, for example, the pure system, it purifies all of these different components this group purified all the different components that are needed to make a protein. And then now it's sold by like NAB. And so you can see it has these like translation factors. It has the tRNA synthetases, so the things that are needed to put the amino acids onto tRNAs. It has the tRNAs, it has the amino acids, it has salts buffers, it has the energy regeneration system, so you could keep the energy going. So it has all of these things that are like purified separately. And so you can see that this would have taken a lot of work. So this group took a lot of work. They had to purify 32 components individually. But when you have this, then you can do things like use non-canonical amino acid incorporation, so you can have them introduce modified protein letters and things. And you can hopefully get more consistent results because the contents are more consistent and various things like that. But you don't have the things that you would have in a lysate, such as chaperones and various things that you might need to help fold. So with the lysate-based system, basically here you're going to have whatever was in the cell cytoplasm. And then this can potentially do things like help the proteins fold. They have things that help the proteins fold, modify them, various things like that. And so these lysates can come from different sources like bacteria, wheat germ, rabbit reticulocyte, HeLa cells, various lysates. And so this is just kind of an example, a real world example of when you might, of some of the benefits of one versus the other. Okay, so moving up towards losing control, but gaining realism, we'll go to in cell culture. And so basically in cell culture, we have this medium level of control. We can introduce compounds more easily than we could in vivo, but less easily than in vitro. So we don't have to worry about it getting into and out of the bloodstream. We don't have to worry about metabolism by the liver. So the liver like kind of changing the molecule, such as making it, this can be either, sometimes it actually toxifies it because the liver is trying to untoxify it, but normally it's trying to like untoxify it and then make it more excretable and things like that, so it can pee it out. But so if it can't get into, if it gets changed by the liver, then it's going to be different when it gets to the cells, if it even gets to the cells. And if it even gets to the cells, then you have to worry about the cell permeability. Can it get through the cell membrane? So they're basically with the in cell culture, you don't have to worry about all that liver stuff, all of that bloodstream stuff, but you do need to worry about the cell permeability, which you didn't need to worry about when we were talking about in vitro. So you can transfect nucleic acids. So that was introducing like drugs and stuff, but you can also transfect nucleic acids. So by transfect, we're basically like sticking it into cells. And there are different methods that we can do this. We can use things like transfection reagents, often like lipid-based reagents that kind of help it get into the cells, viral delivery methods, non-viral methods, like electroporation is often used with these types of cells. So there are different methods that we can use to get nucleic acids into cells, and it's easier to do it this way than when they're actually like in an organism. We cannot, however, tightly control the concentrations of the reaction component. So we have even less control than we had with the lysate base because we're not like adding extra things in. It's harder to change what's already there. So we can't just do that immunoprecipitation or anything. We can use gene editing techniques to generate cell lines with like permanently missing or altered versions of proteins or functional RNAs, et cetera. So with one of these gene editing techniques like CRISPR, you're actually changing the permanent genetic information. This isn't as easy as it sounds, and often what you're doing is when you're trying to make like a knockout, you're targeting a specific gene, but in a way that, and then like having it try to correct itself, and then it messes up, and then the gene is unfunctional. But you have to actually validate that it's actually worked, that you don't have off-target effects, that you're not making the protein that you want. So it's time-consuming and requires a stringent validation. Another alternative is like you can use a gene silencing technique, like small interfering RNA, siRNA mediated RNAi. So I talk about this a lot in my grad school post because I was studying RNAi. And this targets the messenger RNA copy, so the RNA copies of the DNA, and without actually altering the gene. So it's a temporary change, but you can decrease the levels of a specific protein. And then you can actually transfect in the instructions for making different proteins. They're different, depending on your method, you might be able to do it in a way that it'll then get passed on to other cells. So you can generate like stable cell lines that are expressing like a mutated version of a protein or not making a various protein. So you have this more medium level of control. You also have a medium level of realism. So it's more physiological or like bodily realistic concentrations of things. And you have all the intracellular components necessary. However, the expression of various proteins and the concentrations of various components may differ from cell type to cell type, and further depending on cellular stresses, nutrient availability, this sort of thing. You're also lacking the signaling that's coming from other cells, the extracellular matrix, so the stuff around the cells, and various things like that. When you're doing cell culture-based experiments, you're gonna make some decisions. So chiefly is what cell types. So often there are different immortalized cell lines that are used, and these cell lines often come from cancer cells, which allows them to grow for like a really, really long time. But this is because these cells have a bunch of mutations that make it so that they can grow for a very long time. And these mutations, they can have like chromosomal abnormalities. So they might have a different number of chromosomes than other normal cells. And so these might actually impact your experiment. You often need to validate the findings in multiple cell types or cell lines to make sure that your results are more widely applicable. So sometimes you can actually get a patient or experimental animal cells and grow them in cell culture and test them to study the impact of mutations, on the genetic background of the patient or of the animal. Similarly, you can take cells and you can turn them into induced pluripotent stem cells. So basically what happens is that in like a stem cell, it has the ability to turn into or like differentiate into different mature cell types. And so a pluripotent stem cell has the ability to transform into a variety of different cell types, not all of them, but a number of them. what we mean by induced is that you can take a cell and you can actually revert it back to a stem cell-like state. So you can take a cell, for example, from a patient's skin cells and reprogram it back into this induced pluripotent state, using like a mix of different hormones and chemicals and stuff. And then you can add a different mix of hormones and chemicals and stuff to get them to differentiate, so to like mature into a different cell type, such as a neuronal like cell, so type of brain cell. And this is going to have the patient's genetic makeup, but you're not like having to take cells like they're out of the patient's brain. And so you can make it so that you have like very specific types of cells that you want to study. But I know from a little experience in undergrad when I did a summer research internship fellowship in Steven Finkbinder's lab, actually just like right across the street at Gladstone, that this is definitely easier said than done, and these cells take a lot of work. And so some cell types require much more maintenance than others. So you might be spending a lot of time actually just doing cell culture when you're really what you want to do is see what's happening inside of the cells. You also need to think about like what expression scale, wells, plates, flasks, little like circular ball type things. There's various different things to think about. Okay, so now we're moving on to in vivo. So here we're going like inside of organisms. So here we have the least experimental control, but the most realistic, at least for the organism that you studied. As we talked about, it might not be directly transferable, the findings might not be directly transferable to the system that you actually care about. And different organisms are different for better for different recapitulating various aspects of. So one animal might be better for studying one type of like organ in a human, or the other another, and that sort of thing. Also, so in vivo here, so if you're studying like a plant or something, then that's less of an issue. You can actually study the plant that you want, or things like bacteria, yeast, that sort of thing. But remember if they're single cell, it's the same as in cells. Okay, but you basically, so in vivo, you have the least experimental control. So there's natural variability between organisms, and this may influence the results for better or for worse. You have to worry about all of those things. So if you're trying to get like a drug into the cells, it has to go through all of the stuff, surviving the metabolism, getting into the cells that need them where they need them. And so this can be bad in that your drug might not actually be getting into the places that they're needed, but it can also be good because it's telling you that there's a problem getting the drug into places where they're needed, and it might not be going to be work. It's even harder to change things genetic wise than it was in cell culture. Depending on the organism. So again, we're not talking here about like yeast or bacteria, or even like some model organisms like flies and worms and stuff. But if you're doing something with a more like a mammal system or something like that, this is probably going to be like the most expensive, likely the longest time course, highest maintenance, and you might need like institutional review board or IRB approval. And then you have to actually be working with animals if you're working with animals. Yeah, so personally, I leave that work to others, and I'm most comfortable in vitro work, but it is important work. Okay, so another thing is that because of the expense and because of having to sacrifice animals, and it can be hard to get a large enough sample size to make firm statistically sound conclusions. So if you are doing experiments in animals, you have to make sure that you're designing your experiment in such a way that it's robust enough, that you have a large enough sample size that your results are actually going to be meaningful. So you can actually calculate like how big of an effect size you can see with the various population size and that sort of thing. But I'm not a statistician. I know that there are things that exist to do that. You also might be limited in the kind of data you can collect. So is it possible to take samples over time, or does the animal have to actually be sacrificed in order for you to take a sample? In which case you'd have to be getting samples from different animals, then you'd have to have more animals. Whereas if you're just doing something in cells, you can just monitor them over time. if you, or can you just like take cell samples? If it's like in vitro, can you just take little aliquots out? Various things like that. Can you monitor some sort of a reporter over the organism's lifespan? Those are the sort of things that you would be able to do with in vivo system. Is like if you can have some sort of fluorescent reporter in the animal, if you can be taking blood samples, if you can be and checking for various things like that, or growth, or those sort of things that you'd be able to measure. But you're measuring, sorry, you're going to be more limited than you would be in a different system. There's also some stuff in between the cell culture and in vivo. For example, experiments can sometimes be performed in tissues and in organs like ex vivo, so like outside of the actual animal, for better control and easier measurements. Nowadays, cells can be used to grow like organoids. So these are like synthetic masses of cells that have different cell types, etc., that try to recreate a more realistic cellular environment, including the extracellular matrix, various things like that, various signaling messages coming from the different cells. And these are often made from patient cells, so you get the genetic background of the patient. No matter what type of experiment, you're going to want to make sure that when you're planning, you're going to be thinking about your readout. So no matter what your system is, you need to think about how you're going to read out what you're actually trying to study, and make sure that you gather all of the data that you're going to need to answer the question that you want. And this is going to depend on your system and the questions that you're asking. So you can imagine that in an in vitro system, there's fewer questions, fewer data that you could be collecting. And from a single experiment, whereas if you have something happening in vivo, there might be a lot more things that you need to be considering in compounding factors and that sort of thing. So happy designing your experiments, and I hope that helped.